supercomputer. And let me restate that for the recording. So it is the goal of any science to resolve the variation that exists in nature into a singular pattern that can be understood through logical means. Every single science, whether it be physics, chemistry, biology, there is variation that exists in the world. You observe that variation, and what you are trying to do is figure out that variation into a single pattern that we can understand through logical means. Now, you can choose to understand this through a deity, through some kind of religious or spiritual or non-natural means, and that's called religion, and that's fine. That's not what science does. Science tries to understand everything through logical means. So this is why science and religion really don't touch each other, because they're really going after this fundamental question through two very different lenses. Now, the probability of any event is something that quantifies the likely outcome of that event. What is the probability, for example, of the sun rising tomorrow morning? Pretty good, huh? I mean, it's going to be pretty easy to say, yeah, it's going to rise tomorrow morning, except we don't know that it's going to rise tomorrow morning. All right. The probability of any event is never 0% or 100%. It always lies between those two numbers because anything is possible. In fact, the sun will explode at some point. And so at some point, it's not going to rise tomorrow and the world will end. All right. So the probability of any event is never 100%. It can be close to 100%, but it's never going to be 100%. What is the probability of heads? Go ahead, chat it. What's the probability of flipping a coin and getting heads? Scott says 50-50. 0.5. Ah, Sophia asks, is it a fair coin? Great question. Is it a fair coin? Well, sure. Let's just suppose that it is a fair coin, but we don't know if it's a fair coin. And in fact, most coins are not perfectly fair. There's always going to be slight variations, slight dings to them. And so, yes, if the coin is perfectly fair, it's a 50% chance. Here's where things get very philosophical. If we could measure all of the variables influencing the coin, for example, which hand am I using to flip it? How weighted is it on one side? What metal is it made of? Do I start on heads? Do I start on tails? What's the air pressure above my hand? How strong is my thumb? How tired am I when I do this? How do I catch it? Do I catch it with my hand or do I let it fall to the floor? If I could measure all of those variables influencing the coin, feed all of that data into an extraordinarily fast supercomputer, let's say a supercomputer that can do this faster than it would take for the coin to actually flip and then land, could we know with certainty which side it's going to land on? I'm asking. Chat it or say something. Anya says yes. Daniel says no. Brianna says no. Taylor says no. Maybe. Andrew, why do you say maybe?
I feel like because it's not like nothing's like true random. So if you have like all like the information, then maybe you could tell.
So that's right. You're integrating information. So by default, we as scientists reject the idea of randomness. So we have to assume that the world has patterns, which is great. So we like to think that, yes, if we knew all of the variables influencing the coin and we feed that data into a fast supercomputer, we could know it with certainty. However, will we ever know all of the variables influencing the coin? I'm getting no's. So that's more of the important question. I think that if we knew all of the variables, we could know for certainty. And we probably could theorize about a supercomputer that could predict instantaneously which side the coin would land on. Maybe not instantly, maybe it could do it over the course of a day and figure it out through complex computations. However, the caveat is we probably will never be able to measure all of the variables that influence that coin, because there's always going to be something else. The spin of the earth, the size of the universe, everything could influence everything else. And so, although some things are probably not going to influence the coin all that much, we probably can never know everything that influences the coin. So, we are limited as scientists in thinking that, yes, we can probably get at certainty if we knew all of the predictors, but we never know all of the predictors. And so, we're never going to be certain. And so, where does that leave us? It leaves us with a gigantic headache saying that we are never going to know anything for certain. And all of you should be thinking, well, why the fuck am I a scientist then if we're never going to know anything for certain? Well, because of course, some things matter more than others. And so our goal is to understand which of those things matter the most and ignore everything else. One problem that we have in science, in probability theory, is that the simple event of observing a phenomenon is likely to change the probability of that event. Hold on. There we go. Okay. The simple event of observing a phenomenon is likely to change that event. This isn't really a problem. We can incorporate the act of observation into the model. For example, if I hypothesize that insects prefer brightly colored flowers, I don't actually hypothesize that insects prefer brightly colored flowers. I hypothesize that in the presence of a human observer, insects prefer brightly colored flowers. That's always the hidden hypothesis. We never make a hypothesis without that. The hidden hypothesis is always in the presence of an observer. We hypothesize this thing. But because that's always present in the hypothesis, we never include that. But you have to understand, we could, if we wanted to, put a camera trap up. But then in the presence of a camera trap, insects prefer brightly colored flowers. It's kind of it's a catch-22. So we're never going to understand the world outside of what humans observe it to be. So then the question becomes, what are we actually measuring? Are we measuring nature, or are we measuring a human-induced version of nature? And the answer is that we are definitely measuring a human-induced version of nature, because we can never do that elsewise. We can get close to it, though. Some sciences have been able to get closer to it than others. Some sciences have been able to reduce the variability to get the human factor nearly out of it and try to observe nature without any other interference. For example, physics. My colleagues always make the quote that ecologists have physics envy, because physicists have been able to narrow down, whittle away all of the variation that exists in nature. What you're looking at right now is something called a Super Hadron Collider. It is a gigantic magnet, much like the magnet that we talked about in controlling cows and in the early lectures. So this is a gigantic magnet, and in the very, very center of this magnet is a tiny little hole in which electron whizzes through. And so this Large Hadron Collider is actually 18 kilometers long and borders 2 different countries in Europe. And its entire purpose is to accelerate magnets. Anna says, I'm having trouble getting the notes without being able to pause like normal. Anna, I will post the lecture after class. Actually, you know what? Let me get the lecture posted right now. I'm sorry that I didn't do that. That's okay. Oh, shoot, I have to close it. Hold on. Okay, it's posted on Blackboard. Thanks for letting me know that I forgot to post it. And let me reopen PowerPoint and get that slide back up for you. Okay, so this is a Large Hadron Collider, and it is designed to whiz electrons through at nearly the speed of light in the absence of any humans to see what happens when you smash them open together. So this is understanding, this is the thing that if you recall, maybe 5-6 years ago, they were worried about a black hole opening it up in Europe. This was that thing. And so they're trying to smash electrons into each other. And they've taken a lot of variation out of nature, right? But you have to understand that physics has had a long time to work out the variation, to figure out how to observe nature. When Galileo first looked at the stars through a telescope, there was a lot more variation than they knew what to deal with. And so they were very confused by what is happening. They did not know how light bends. They didn't know how far even the sun was. They didn't know any of this stuff. We over time as a society have figured out a lot of this variation, and we've also figured out how humans influence all of those variations a little bit, and so we can narrow away the variation. Ecology and environmental science in general as a field is only about 100 years old, and so we are still working with a lot of variation. Now think about understanding when a seed germinates. We still can't figure out within, like, I don't know, a couple days when a seed would germinate. It takes some time. You know, sometimes seeds just don't germinate, and we accept that. We don't know why. We're not really sure. I mean, we're just chalk it up to some genetics, but some sciences have been able to work around that variation. Ecology, not so much. And so ecology has a tremendous amount of variation still. And this is one of the reasons why we insist that students take statistics, is because variation is so prevalent. Physicists, chemists, they actually don't do much statistics at all because there isn't a whole lot of variation. If they see variation, it's usually because they fucked up an experiment. In ecology, there is so much variation. If we don't figure a way out to resolve some of that variation with statistics, we're screwed. All right, so the probability of anything is probably, depending upon whether or not you can think about whether or not we could actually get all of the variables predicted. The probability of anything is between 0 and 1. It's not going to be 0 and 1. Probability of anything is going to be between 0 and 1. All right, nature is incredibly variable. To give you like a biological example to work with, I just want to show you guys a video. What I'm going to do is I'm going to copy and paste this link into the chat so you guys can check it out. All right, let's take a minute. Let's watch this video together, and then we'll come back. Okay, it's in.
If you work in a team, you've got to try Monday.com. Go to Monday.com and manage all your team's work on a powerful platform that's easy to use. So easy. Whether it's projects or workflows.
It's said sometimes it takes a wrong turn to get you to the right place. But landing up in this nightmarish neighborhood is purely a stroke of bad luck. For these Venus flytraps are hungry for flesh. With just a touch to one, and then two of the microscopic hair triggers, the plant's jaws trap the prey, setting in motion a bizarre all-in-one digestive system. The sealed pod floods with a lethal brew of digestive juices. Its mouth becomes its stomach, and finally its intestines, as it absorbs the nutrients. But this isn't the most efficient system, for a missed meal means energy wasted. High up above the flytraps, fellow carnivores, the pitcher plants, are a little more proficient. They're passive hunters firmly rooted in one spot. These plants make no attempt to hide in ambush. Once on the lip, the insect is as good as dead. Downward-facing hairs prevent a good foothold. The toxic nectar drugs them. Deep, waxy walls make it too slippery to crawl out. Others are also fighting for their lives. All the while, without their knowing, they're already part of the digestive process. Their remains will soon become part of the existing graveyard. The greatest guzzlers belong to the tropical pitcher family. Sporting deep tubular pits filled with flesh-dissolving enzymes, it's like an open-ended stomach. Any animal small enough to fit through the mouth of the death pit is fair game. Victims die by drowning, while their flesh slowly dissolves.
Okay, so you have the gist of it. These pitcher plants are super, super cool. And we have some of them actually in southern New Jersey. Southern New Jersey Pine Barrens is actually a hot spot for carnivorous plants. We have several different species of them, including sundews as well as pitcher plants. I even have some in my office just as my pet little pitcher plant that takes care of any bugs in my office. All right, so now that we have this idea in our head, I just want to let you know that there are some organisms like frogs that actually live in these pitcher plants. They consume insects, and in some tropical systems, frogs will actually lay their eggs in the pitcher plants. The tadpoles have these evolutionary adaptations where they don't suffer as a result of the enzymes in there. They actually treat these as a home. So what is the probability of an insect visiting a pitcher plant but not going in, visiting, going in, and escaping, and capture? Let's assume that there's only 3 things that could happen. Let's assume that it's one of these 3 things. All right, nothing else could happen. No other probability. So insect visiting a pitcher plant, it goes in. It does not go in. It visits, goes in, and then it escapes. And then what is the probability of an insect going in, getting captured, not leaving? There are 3 possible events. And the way that we denote those 3 possible events is in something that we call probability space, which is, it's denoted by these curly brackets. If you ever wondered why we use these curly brackets, here it is. These curly brackets indicate a set, if you will, a set of events. And so the probability of the insect doing something, in other words, the insect visiting, is 100%. Within that 100%, there's 3 different possible events that add up to 100%. What is the probability of hovering but not entering, entering and leaving, and entering and staying? So we observe 100 events. Let's say that I'm interested in knowing how efficient pitcher plants are at capturing. And so what I do is I go to the Pine Barrens and I find a pitcher plant and I watch the pitcher plant and I watch 100 insects visit the pitcher plant. I don't know what those insects are. They could be mosquitoes, they could be gnats, whatever. We watch 100 events on one pitcher plant. And on 50 events, the fly hovers but does not enter the pitcher plant. On 40 events, the fly enters and then leaves. And then on 10 events, the fly enters and does not leave. So we say that the probability of hovering but not entering is 0.5. It happened 50 of those times, so it's 50%. The probability of entering and leaving is 40%. The probability of entering and capturing is 10%. If you forgot that percentages are denoted as decimal points, please remind yourself of that now, because we're going to be using that for a lot later on. 50% is denoted as 0.5, 40% is 0.4, and 10% is 0.1, 100% is 1. The sum of all of your events in your sample space must equal to 100% or 1. It has to. And if you go back here, you can see that 50 plus 40 plus 10 is equal to 100%, or 0.5 plus 0.4 plus 0.1 is equal to 1. If you define all of the possible events that could occur, your sample space must equal to 1. Now, I want you to keep in mind that this is an observation of one plant. And so in such an experiment, your conclusions are limited to that one plant, which is a bad experimental design. If I want to know generally the capture efficiency of carnivorous plants, and I only observe one plant, I can't really say anything in general. I just know about that one plant. So what we're going to do instead is we are going to observe 100 insects that approach 20 different plants. All right, how do I do this? I recruit a whole bunch of undergraduates. Let's say I recruit 19 undergraduates and I include myself, and we all go out to the field and we all find a pitcher plant. This beautiful, wonderful day, we all go out there, we socially distance, and we all sit our butts down on the ground, and we all just watch our pet pitcher plant. And we say, okay, you cannot get up until you observed 100 insects approaching that pitcher plant. So collectively, we're going to observe a total of 2,000 insects. Got it? So in that case, what we are going to do is collect all of our data together at the end of the day. And let's say that one person, let's say I observe 50 insects hovering but not entering my pitcher plant. And then I go to Sophia and I say, what was your data like? And she says, oh, my data, I saw 47 insects hovering but not entering that pitcher plant. And then I go to Anya and she says, oh, I saw 37. All right. I go to Abby and she says, oh, I saw 53. All right. I go to Scott and he says, oh, I saw 65. All right. So you keep on doing that and you poll everybody. And so you keep on adding numbers. Now, if I put numbers on top of here, it means that somebody else in the class also observed 50 insects hovering but not entering. And somebody else also observed 53 insects hovering but not entering. We keep on collecting our data and we keep on saying, okay, well, how many insects did you observe hovering but not entering? We keep on doing this, keep on doing this, keep on doing this. And what we end up with is a histogram. A histogram tells us what is the frequency of that observation. Now, 5 students found 50 insects hovering but not entering. Only 1 student found 27 insects hovering but not entering. Only 1 student found 71 insects hovering but not entering. So 71 and 27 are fairly rare events, and the majority of events seem to occur somewhere between 47 and 53. This is a histogram. In our next class, we will have a practice exercise where we develop histograms and we show you how to manipulate histograms, but I need to introduce you to what a histogram is first. We could arrive at this distribution a number of different ways. We could, if we wanted to, measure one plant at different times. So let's say that I go out one day and I measure that one plant and I see 50 insects hovering but not entering. I go out the next day and I see 27 insects hovering but not entering. I go out on day 3 and I observe 71, and I go out day 4 and I observe 50 again. And I go on for another 20 plants and I observe 100 insects at a time. This would be your trajectory experiment where you are observing one organism over time. Now, this isn't ideal because, of course, you're only getting data for one plant, and so you cannot generalize about pitcher plants in general. You only know what's going on with that one plant. This is very much like observing your one pet and assuming that your pet acts like all other pets. I know my cat does not act like all other cats. And so if I made an observation about my cat, and if I made a histogram about my cat, that's not going to be representative. And so it would be much better to measure 20 different plants. We could also measure 100 insects at multiple plants at one time, which is what we did in that previous experiment. This is known as a snapshot experiment. Instead of following one plant over a long period of time, we take a snapshot of a forest and look at the pitcher plants in that forest. So we measured 20 plants at one time and 100 insects at each one of those plants. All right, so we could do that via a trajectory where we measure one over time, or a snapshot where we measure 100 insects over time. The best thing that we could possibly do is measure a trajectory and a snapshot, measure 20 plants over, I don't know, 20 different days. That would be really cool, but we often don't do that. But like I said, if we measured a single plant, this changes our conclusions. All right, this is nothing new. I just kept on saying this before. All right. If we measure a single insect at 100 plants, this would severely change our conclusions because this changes the entire focus of the experiment to the insect instead of the plant. If I follow an insect around, this is a weird image for your heads. If I am there and I'm sitting in a forest and I'm looking at that insect and I follow that insect around to a whole bunch of different pitcher plants, that's going to severely change the focus of this experiment because, of course, that insect is very, very good at avoiding getting captured. So this isn't particularly good if my question is, how efficient are pitcher plants at capturing insects? All right, let's suppose that we did measure 100 visits at each plant, but instead of 100 plants, we only measured 4 plants. All right. Or I said instead of 20 plants, I think before we only did 20 plants. Instead of 20 plants, we only measured 4 plants. What will our histogram look like? It's going to look the same, sort of. Our first data point was 50. Our next data point was 65. Our next data point was 27. Our next data point is 41, and we stop there. It doesn't look like a very nice histogram. However, in the bulk of experiments that we do, we don't take a whole lot of measurements. It's kind of hard to sit there and measure pitcher plant after pitcher plant after pitcher plant. And I'm guessing that I'm not going to be able to recruit 20 of you to go out to a forest at one time. So this is still, what you're staring at is still a histogram. Even though there are 4 points, it's still a histogram. And it still has a property that the bulk of data would occur right here. Now, if we assume certain things, and I want you to hold on to this assumption, if we assume certain things, if we assume that the greatest number of points would be near the average, near the middle here, so that if we kept on adding points to this, we got a hump right here in this area right here, we assume that the data would follow what we call a normal distribution. I want you to hold on to that normal distribution thought because we're going to dwell on that more in a later lecture. This is what a normal distribution will look like if we assumed that the majority of data points would be right here. And in a lot of cases, we have to assume that. A lot of my experiments, we only have 4 replicates. because more replicates is just not possible to do. I mean, in these experiments where I fill up lakes, where I fill up cattle tanks with water, filling up more than 4 lakes with water is logistically almost impossible to do. And so usually we only have 4 points, and so we have to assume that there is an average right at the center of all of this data. All right. So no, it doesn't look like much of a histogram, but if we assume that the average would represent the most commonly measured probability, then we can still estimate the average to be right around the middle of these data points. Please do chime in if you have any questions, all right? Or if you want me to repeat anything, I am more than happy to. I'll take a drink of water. All right. I want you to note also, we have not measured all events. Let's suppose that we did measure 20 pitcher plants. Let's suppose that we measured 100 pitcher plants in a forest. We still haven't measured all of the pitcher plants in the forest. And if we have, we still haven't measured all of the pitcher plants in New Jersey. So we say that we have taken a sample of insects visiting pitcher plants. This is very important. So if you don't get this, please let me know. We have just taken a sample, and we hope that our sample is representative of the rest of everything in the world. If you just measured the activity of your household cat, you are not really justified in assuming that your household cat represents the truth of the entire world. If you measured all of the cats in your neighborhood, you're getting closer to being able to correctly estimate the truth based on your sample of cats. And likewise, if I measured all of the insects visiting all of the pitcher plants in the forest, I could get much closer to truth, although I still will have not measured all of the pitcher plants throughout all of time. The more replicates I have, the better I have the ability to estimate truth. All right. For each trial, let's change this example up a little bit. For each trial of 100 insects, I record the type of insects that are visiting the pitcher plant. Okay. So let's suppose that there's 4 types of insects that visit the pitcher plant. 20% of them are mosquitoes. Okay. 30% of them are gnats. Yes, I know the G is silent, but I like saying gnats because it should be said gnats. Also, I like saying herbs because there's a fucking H in the word. And if you all know that that's from Eddie Izzard, you're my friend. So there are 10% fruit flies, 20% midges. Midges are awful. Midges are just like biting insects that are awful. Okay. So 20%, 30%, 10%, 20%. That doesn't equal up to 100%, Aaron. What the hell is going on? Okay, fine. You know what? Realistically, 20% are going to be unidentified. All right. You're watching them whiz around. So let's make this example real. 20% of them, you're just not going to know what the hell they are. And so 20% on a good day, unidentified. Cool. All right, guys, don't be scared. I'm going to introduce some math for the first time, I think, in this class. If the probability, and these should actually be squiggly brackets. I'm sorry. These should be squiggly brackets. Go along with the notation here. If the probability of mosquito visiting the pitcher plant is 20%, and if the probability of the pitcher plant capturing the mosquito is 10%, then the probability of a pitcher plant getting visited and capturing a mosquito is equal to 20% times 10%, which is 0.2 times 0.1, which is 0.02 or 2%. I'm gonna try to repeat that in a slightly different way. What we're doing, we sit down and we observe pitcher plants. And we are simultaneously observing how many of any insect visits and leaves, hovers, enters, and then leaves, hovers, enters, and is captured. And we know that the probability of the pitcher plant capturing any insect, regardless of what type it is 10%. Now we know that 20% of the insects that the pitcher plant actually gets visited by are mosquitoes. So the probability of it being visited by a mosquito and the pitcher plant actually capturing that is 20% times 10%, or 2%. Likewise, if the probability of getting visited by a gnat is 30%, and the probability of the pitcher plant capturing an insect, it hasn't changed. That's still 10%, but the probability of it getting visited by a gnat is 30%. And the probability of getting visited by a gnat and capturing that gnat is 0.3 times 0.1, or 3%. The probability of a fruit fly visiting is 10%. The probability of that fruit fly being captured is again 10%. And so the probability of fruit fly and capture is 0.1 times 0.1, or 1%. Midges, same thing, 20%. The probability of capture is 0.1. 0.2 times 0.1 is 2%. And of course, the last one, the probability of the insect being unidentified is 20%. The probability of capture is 10%. So the probability of the insect being captured that is unidentified is also 0.2 times 0.1, or 2%. And you're saying, wait a second, those are really small numbers. You're right, those are really small numbers. How in the hell do we get up to 100%? Hold on one second, because think about this. Let me go back to this first slide. We know that the total probability of an insect being captured is 10%, which means that the probability of a mosquito being captured, plus the probability of a gnat being captured, plus the probability of a fruit fly being captured, plus the probability of a midge being captured, plus the probability of an unidentified insect being captured has to equal up to 10%. Has to equal up to the total number of captured insects. Hope that didn't confuse you, because I can see how it might. I'm going to go on, and if that did confuse you, hopefully the next couple slides will help that out. All right. Oh, that's exactly what I just said. I could have just gone to this slide if I just remember what my slides are. The probability of a gnat being captured, or a mosquito being captured, or a fruit fly being captured, or a midge being captured, or an unidentified insect being captured is equal to the sum of all of those percentages. So at this point, I want you to remember something. Jot it down if you're taking notes. If you're measuring the probability of something and something else happening, you multiply the two numbers. If you're measuring the probability of something happening or something else happening, you add the two numbers. Did I say that right?
Can you repeat that?
I think I might have said that wrong. So absolutely, yes, I'm going to repeat it. The probability of something happening and something else happening together. Let's say the probability of you rolling a 1 on a fair die and then rolling another 1 on a fair die is equal to the product. Sorry, I think I said it wrong the first time. It's equal to the product. So you multiply those two. The product of something or something else happening is equal to the sum of those two probabilities. I'm going to say it a third time because I know I said it wrong the first time. The probability of two things happening together, so something happening and something else happening, is a product. You multiply them. The product of something happening or something else happening is the sum. I'm going to go through another example that's going to solidify that. Okay. God, why don't I remember what slides I have in here? Fucking A. Right, that's exactly what I just said. The probability of two events occurring together is the product of the probability of each of the events. The product of the probability. Versus the probability of one or a second event occurring is the sum of the probability of each of those events. Let's bring this home with a fair-sided die. All right, something that everybody can associate with, because at some point you played Monopoly or something else, or you gambled, I don't know. All right, so let's say you have 2 die. 2 dice? 2 dice? Yeah, you have 2 dice. I don't know, whatever. All right. What the fucking hell? I wish I remember what my slides were. Okay. What is the probability of rolling a 6? It should be 1 out of 6. What is the probability of rolling another 6? Again, 1 out of 6. What is the probability of rolling a 6 first and then another 6 after that? It is 1/6 times 1/6, which is 1/36. The probability of rolling 2 6s in a row is equal to the product of the individual probabilities, which is 1/6 times 1/6. So 1/36, which gets you, and if the probability of rolling a 6 a third time would be 1/6 times 1/6 times 1/6, which is equal to, I don't know, 100 and 200 and something. I don't know. Do the math. Somebody tell me. So what is 6 times 6 times 6? Somebody.
216.
So rolling 6 3 times is 1 out of 216 chances. Okay. Now, what is the probability of rolling either? Let me go back to this one. Sorry. What is the probability of rolling either a 1 or a 2 or a 3 or a 4 or a 5 or a 6? Somebody. 6 out of 6, which is 1, 100%. When I say or, what is the probability of rolling a 1 or a 2? Then we say it's 1/6 plus 1/6, which is 2/6, or simplified as 1/3. So the probability of rolling a 1 or a 2 would be 1/3. The probability of rolling some number on a die is 100%. The probability of rolling numbers in a sequence is equal to the probabilities of each of those numbers times each of their events. So the product. All right. What I want everyone to do now, of course, if anybody has any questions or you want me to repeat anything, please let me know. There is a quiz on Blackboard. It's under the quizzes, it's under the quiz section, but it's labeled for in class. It's a quick practice in those probabilities. I'm going to put everybody into breakout rooms, and I want you guys to go through that quiz together. And I want you to calculate what you think the probabilities are. Now, be careful and pay attention to the wording. All right. I have got to go check on the fire to make sure that nobody's dead. Although I'm guessing that somebody would have texted me by now if somebody were dead. In any case, that's what I'm going to ask you guys to do. If you get 100% on that quiz and you don't have any questions, you're done. You're free to go. But I do want to make sure that you guys understand the rules of probability before I let you go.